Okay, hello everybody. I'm Sharon. So I've had Leonbergers for a little over 25 years, and I grew up with some German Shepherds, and I've been breeding for 20 years now. I became involved in the international Leonberger scene about 11 or 12 years ago. I was asked by the then president of the International Leonberger Union to do a fertility study on the breed, which we did. And subsequently, the first health group for the ILU was founded, and I was involved with that. And we produced a video about genetic decline in the breed. I have become a member of the board of the Leonberger Health Foundation since then. I'm a past member of the committee of the Leonberger Club of Great Britain, and also their health committee for a little over a dozen years, but I stood down from those posts a few years ago. And I'm now the Kennel Club, the UK Kennel Club's breed health coordinator for Leonbergers here in Britain. So that's who I am. So before I actually begin, I want to just say that by now, many of you have probably heard about the proposed Leonberger outcrossing project called LEOGEN, but you may have also heard about another unrelated group that has already crossed a Leonberger with an Estrella. And I just want to say right now at the outset that this is entirely separate from us. We have nothing to do with that, and it's an entirely separate effort. And I'm sure that everybody has their own views about all of this, but what I want to do today is to try and present the reasoning behind the proposed LEOGEN project, which is a proposal. It hasn't started yet. And I want to present this in terms of some very clear and simple questions. So the questions being, we're okay? Why, who, how, and then take questions at the end. So let me see if this will work. There we go. Why? So in the autumn of 2020, a comprehensive summary detailing the genetic health of our breed was published. If you have been involved in Leonberger health matters for any time now, you will probably recognize the names of many of those experts who co-authored the study. Anna Letko and Cor Drogmuller from the Institute of Genetics at the University of Bern, Katie Miner and James Mickelson from the Canine Genetics Lab at the University of Minnesota, Peter Olioch, the conservation geneticist from Dogs Global, who did the presentation of the 2016 video Safeguarding Genetic Diversity in the Leonberger Breed, and others. So together, these authors represent the top genetic specialists in our breed. If anybody knows about where we are as a breed, it's them. The report that they put together in 2020 presents the current data on the genetic state of our breed, and I encourage anyone with a serious interest in the future of this magnificent breed to read it, because it is very, very concerning. So one key finding showed that today's Leonbergers has an average coefficient of inbreeding of 30% when all generations are taken into account. And remember that a brother-sister mating will produce a 25% inbreeding coefficient. This figure of 30% is based on both theoretical calculations derived from pedigree analysis, but it's also derived from direct molecular DNA analysis. So there's no question of parentage mistakes along the way. The results were both essentially the same, and together they verify an inbreeding level of around 30% overall. And most of you, I hope, will also be familiar with the mean kinship analysis that was initially conducted on Leonbergers back in 2015 by Dr. Peter Olioch over in Holland. This demonstrated a similar level of relatedness, as well as pointing out the shockingly low founder genome equivalent that we have remaining in our breed. And what that is, the founder genome equivalent represents the remaining genetic contribution from the original founders of the breed that, if mated together, would produce the same diversity, and thus the average mean kinship as we now have in the current population. In the Leonberger today, this number is apparently just 1.59, meaning that we now have less diversity in our breed overall, than that which would come from two unrelated founder dogs. The total mean kinship calculation for our worldwide breeding population has now been done 3 times. And each time, the result shows a clearly worsening trend. The most recent calculation of mean kinship levels shows us that there has been a further reduction of some 2% in the diversity of our breed over just the previous 2 to 3 years. The most recent figures from the worldwide independent database of Leonberger pedigrees also shows that the level of inbreeding continues to rise, despite the fact that overall breeding population is relatively numerous. In other words, we're not a rare breed. And this indicates that the problem of eroding genetic diversity is essentially baked into the breed. In other words, within the fixed boundaries of the current closed studbook system, there will only ever be less, never more, diversity available to future generations. And this applies to all breeds within the closed studbook system, not just the Leonbergers. None of this is speculation or rumor. This is what the science is telling us. So, what does this mean for our Leonbergers? Livestock breeders for many decades now know the answer only too well. Inbreeding depression leads to poor health, less fertility, shorter lifespans. In some countries, such as Germany, annual figures are available for both the number of matings done and the number of actual litters that are produced. These show that the conception failure rates have been as much as 40% in recent years. Health data on Leonbergers collected over many years shows us that the average lifespans are reducing. To quote the 2020 report that I mentioned earlier, based on a study of more than 3,000 records, a slow, continuous decrease from an average of 9.4 years in 1989 to 7.7 years in 2004 was observed. The Finnish Kennel Club data from 10 years later shows average lifespan is recorded as 7.5 years. Studies in other breeds have indicated that the higher the coefficient of inbreeding overall, the lower the predicted lifespan will be. Some studies even suggesting 1 to 2 years on average drop for each 10% jump in coefficient of inbreeding. So I could go on, but I'm sure, get the idea. So now many may be saying it's not just our breed that shows a dangerously high level of inbreeding. Many pedigree breeds also show this, which is certainly true. So you may ask, why should we feel that we must take such a radical step now as potentially crossing our breed with others to bring in more diversity? Yes, there are many breeds with similar problems caused by the closed studbook system. For example, some studies have shown cancer death rates of 55% in Bernese Mountain Dogs and over 40% in flat-coated retrievers. Over 50% of boxers are thought to carry the gene for arrhythmic right ventricular cardiomyopathy. And according to one study, a shocking 60% of golden retrievers will die of cancer, and an estimated 58% of American Dobermans were also found to be affected with some form of DCM. So yes, there are doubtless other breeds as bad, if not worse, than ours in this case. But our question is this: should we wait another 10 or 20 years before we do something about this and start to act? Why should we do that? And I won't go into the specific details of illnesses in Leonbergers that many of you already know, beyond saying that in addition to the shocking cancer rates, the DCM, the neurological conditions that we have seen for many years, we have also now seen new research recently initiated in response to conditions as diverse as sudden cardiac death, pectin ligament dysplasia in eyes, juvenile renal disease, and so forth. And contrary to what some people apparently choose to believe, researchers do not invent new illnesses to study in breeds. Rather, they respond to rising issues. And that is what is happening in our breed. So back to the question, why do this now? Some will say that before we take such an apparently radical step, such as an outcross project, we should instead utilize the existing tools that we already have to slow down this loss of diversity. Because remember, we can only ever We can never reverse a lack of diversity within our closed population. We can only slow it down. What we have is what we have. All we can ever hope to do is to slow down the decline, but we will never stop it. So why not start there, you ask, with the tools that we have, which is a completely sensible question. But the truth is that it has been 12 years since the issue of poor fertility and the possibility of inbreeding depression was raised by the fertility study that I was commissioned to do for the International Leonberger Union, and nothing has changed. It has been 9 years since these problems were so clearly raised with the video Safeguarding Genetic Diversity in the Leonberger Breed. And during these 9 years, breeders have had the tools to change direction. They have had the automatic coefficient of inbreeding calculation tools for 5, 10, and all generations. They have had the automatic test pedigree generators. They have had the mean kinship calculation numbers. They have had the health survey results published again and again. And they have seen new health issues continue to arise, and yet nothing has changed. None of it has reversed the trend that our breed is currently following. These tools simply are not working. So is it now time to try something else? I have been involved in Leonbergers for 25 years now, as I mentioned, and I know that there has been talk about a diversity project for many of these years. Making any sort of meaningful difference will take time, so we are proposing that we start now rather than waiting another 25 years. And please remember that this will not be the first time that outcrossing has been done in our breed. It has been done before in 1954 with Alma von Rossbach, whose mother was a Newfoundland, and who most modern Leonbergers today go directly back to. And it did not ruin the breed, nor have crosses with other breeds that we have shown examples of on the LEOGEN website ruin those breeds. In fact, it has improved them. So why not start now? And let me make one more final point before I move on to who. We believe that it is also important for Leonberger breeders, and indeed all pedigree dog breeders, to be mindful of the bigger picture, namely a rising sense of antagonism towards pedigree dogs among the general public. There is an ongoing decline in pedigree registration numbers. We see it in Britain, we see it in America and elsewhere. There is increased scrutiny of breeding practices, and now, in some cases, examples of direct challenges towards pedigree breeding, such as the recent ban on bulldogs in Norway and the so-called torture breeding regulations in Germany. Perhaps this is also a reason for the rise in designer crosses. Simply put, many people believe that they are healthier. Now, I'm not saying that they are or are not. However, one need only look at the lifespans and cancer rates in so many of our breeds to understand why people might be seeking alternatives. The massive Vet Compass study that was recently published in the UK put the average age of crossbreeds at 11.7 years, whereas the most popular of the pedigree breeds, the French Bulldog, had an average age at death of just 4.5 years. The details don't matter when that is the headlines that people are reading. Pedigree dogs are losing the public relations battle. Dog owners are increasingly turning against the whole idea. There is no point in simply getting mad about this. Instead, we should try and understand why this is happening and to be prepared to show a willingness to look at our own breeding practices and the improvements that we could make. And what do I mean by this? Pedigree dog breeding was born in the Victorian age. The concept was simple. Put a circle around a group of selected dogs and then only breed from within that closely defined group, putting the best to the best and throwing out the rest with each successive generation. Now, this undoubtedly worked very well for 5, 10, 15 generations perhaps, but we are now at 50 plus generations of Leonbergers, and if we continue eliminating over 80% of the individuals from each successive generation and chasing down the same top show winning lines over and over again, it can only lead to one thing, and that is a genetic dead end. We simply can't keep doing the same thing and expect nothing to change, because it is changing all of the time, changing for the worse. And it will keep doing so unless we change our approach. And that's why we are proposing that we might consider doing a project such as this. So the next question obviously is who. So our starting point was the recognition that in order to have a reasonable chance of success, we would need 4 things: collaboration with appropriately qualified scientific expertise, support and reasonable representation of a portion of the Leonberger community, acceptance and support of the kennel clubs, who are the registry bodies that govern our sport, without which any project would be futile. And finally, a suitable DNA profiling lab to help us to identify and track our new genetic contributions. We are now in the age of molecular genetics, and we should use these tools. So our starting point, therefore, was to contact the lead author of the 2020 paper, Genomic Diversity and Population Structure of the Leonberger Dog Breed, which I mentioned at the top of this presentation, Dr. Anna Letko. And she kindly agreed to join us and has become a core member of the team. We're also thankful to now have the support of Professor Cord Drogmüller from the Institute of Genetics at the University of Bern. And they have, of course, been deeply involved with our breed for many years now. Our second step was to secure expressions of support from at least 3 respected national Leonberger clubs who would endorse the project and stand behind the 3 breeder teams formed to carry it out. We have this now from the Swiss, the British, and the Finnish Leonberger clubs. Next, we understood clearly from the outset that we would need the support and cooperation from the registry bodies themselves, in other words, the kennel clubs, because if the progeny that resulted from the LEOGEN project could not be registered, then the breeders of the future could not use them, and that would be pointless. We have achieved strong initial statements of support from both the UK Kennel Club and the Finnish Kennel Club, which is sufficient to proceed with developing the project. In the case of the Swiss Kennel Club, they have expressed a desire to see the project pursued in association with a strong academic partner, and we are very pleased to have such an expression of support now in hand from Professor Drogmüller and the Institute of Genetics at the University of Bern. Finally, we have been negotiating with Embark, a global leader in canine genetics, who hold what is probably the largest canine DNA repository in the world, and have developed a high-resolution genotyping platform that analyzes a dog's genome at over 230,000 different locations, allowing them to screen for literally hundreds of traits, both beneficial and deleterious. Embark is a research partner of Cornell University College of Veterinary Medicine and has what is perhaps the most comprehensive canine DNA bank in the world, including pedigree, dogs, and many, many more from around the world, from village populations, from feral populations, and so forth. So they really have the broadest understanding of canine DNA. And we are pleased to say that they have now presented the LEOGEN team with a proposed outline of ongoing support for each stage of the project. In 2022, there were 4 staged presentations made to successive subcommittees, and finally the full committees, culminating in a presentation to the board of directors of the British Kennel Club. And following this, we received notification that the board were in agreement with the venture and supported the proposal. After this, our colleagues in Finland presented the project proposal to the Finnish Kennel Club, who likewise agreed in principle, and they have a strong track record in these sorts of projects. And the door has now been opened to more detailed negotiations to proceed between the 2 registry bodies, negotiations which will be necessary in the long run for the progeny of the LEOGEN breeding program to be accepted by both kennel clubs in the future, the FCI-based Finnish club and the British Kennel Club. We have also presented an outline of the proposed project to the Leonberger Health Foundation, whose stated mission is to, quote, protect the health, longevity, and preservation of the breed. And we are thankful to have also received a strong statement of support for the goals of the LEOGEN project from them. We have now made 2 presentations to the delegates of the International Leonberger Union about this project and have been encouraged by the subsequent discussions there. So before we begin, we will also have in place an advisory panel, which we expect will include reproductive specialists, specialist judges, geneticists, experienced breeders, and kennel club representatives. And preliminary contact with such qualified experts has already started. So that's the basic groundwork of what we've done. So then the question arises, well, what are we actually going to do and how are we going to do it? And of course, the first question always is, what breeds are you going to use? And this is usually the point where the whole discussion falls to pieces, as we have seen recently so spectacularly on Facebook. However, we believe that the question is wrong as a starting point, and instead, we should be asking ourselves, what do we need? What are the most likely contributions that we could have that would impact positively the health and longevity of our breed? And where can we find those? And that's where we think we need to start looking. So we're not starting with the question of what breed are you looking for, because we're not trying to recreate the Leonberger. We're trying to inject more diversity into the breed and then bring it back to type. So, for example, it would be perhaps beneficial to the immune systems of our future Leonbergers to introduce more variety in DLA haplotypes. And these are what are called major histocompatibility complexes in human genetics and refer to bundles of genes called haplotypes that are inherited as a group and which could potentially add robustness to their immune systems and thus lead to direct improvement in health and longevity. So it makes sense to seek out genetic donors that bring novel DLA haplotypes into the program. Secondly, we know from focused specific research that our breed is very low in maternal or mitochondrial DNA. So of a total of 39 Leonbergers analyzed, only 2 mitochondrial haplotypes were discovered. And of those, all but 1 dog had only 1 version. So in a breed such as the Estrella, by comparison, there are something like 6 distinct mitochondrial haplotypes present, which is a very broad range. With a very broad range mathematical combination, you could end up with over 1,900 possibilities, if my maths are correct, which is the total possibilities for 6 distinct objects to be paired. And there are many more mitochondrial haplotypes found in dogs generally. So, and I'm not saying we're going to use an Estrella, so please don't jump to conclusions. That's just one example. So one of our first genetic donors could perhaps be a female dog, rather than just seeking out semen from donor dogs. And she would carry novel mitochondrial haplotypes into our breed. And these go back, this is the maternal line. This goes right back to the metabolic rate. Mitochondria are existing in every cell, and they are the engines of the cells. So this could make a huge difference. Thirdly, we need to do what we can to ensure that we are not introducing excludable genetic illnesses. And here we are grateful to potentially have a partner such as Embark, who can screen for well over 200 known diseases. This is a common criticism. Well, you don't know what you're bringing in. Well, we can screen out quite a few things. So the common objection that such a project will invariably risk introducing new genetic illnesses is greatly reduced. It is worth pointing out at this stage that they can screen for other factors, such as size, build, color, mass, and so forth, which could be very useful. Finally, doing an ambitious initial introduction of new genetic material is rendered largely pointless if, over the course of a handful of generations, that new genetic material is not tracked and could therefore be quickly lost again. So we are thankful that our potential partner lab, Embark, fully understands this and agrees with the need to track each generation in order to make an effort to, as much as possible, retain the novel contributions, especially in terms of those areas mentioned above. Of course, each subsequent generation of puppies will need to be carefully evaluated and screened prior to selection for the next generation, and this would certainly include all of the present pre-breeding tests, such as for hips, as well as for careful consideration of type. But it is arguable that no quality is as important as temperament. Leos are known for their calm, kind, and confident characters. although, let's be honest, many already do fall short in this regard. And here we have no genetic test. Here too, the argument of nature versus nurture rages as much as it ever has. Therefore, careful selection of guardian homes and temperament evaluations prior to selection will be done. And in a sense, this is as much and no more than breeders today already have to help them in this regard. And here I'd like to take a moment to speak to a specific concern that everybody seems to have, namely that we will be forever changing the phenotype of the Leonberger in our effort to improve the genotype. So phenotype being the observable physical elements and to a certain extent character that define a Leonberger according to the standard, and genotype being the underlying genetic makeup. So let me digress to an example here. There was once a boxer breeder by the name of Dr. Bruce Cantinaw, and he was also an outstanding scientist who led the mammalian genetics unit at the Harwell Institute. He was an exhibitor and breeder of pedigree boxers, and in 1992, he decided to try an experiment and crossed a boxer bitch with a naturally bobtailed corgi. He knew that the bobtail was caused by a single dominant gene. In the initial litter, 5 of the 7 pups had the bobtails of various lengths. Of course, they looked awful, as you can see from the F1 example on the left of your screen. But one of those was selected and back-crossed to a boxer male, and a daughter of this mating, who you can see on the right there, called Jane, was clearly a boxer in her phenotype, and she was selected from there to breed on from. In Dr. Cantinaw's own words, he related that, quote, Jane, as she was to be called, looked very like a boxer, both as a small pup and subsequently when she reached normal boxer size. And her temperament was delightful, being boxer in most ways, if perhaps more sweet and gentle. In effect, we had got our boxer back in the first back cross generation. The project continued for many generations, and the progeny were registered eventually and competed at the highest levels. There are many other examples, but this one is most impactful in demonstrating that phenotype can be recovered, and often relatively quickly, even in such a diverse breeding. And it is in no way certain, as some would have it, that an outcross will ruin a breed. Please remember once again that Alma von Rossbach was an outcross, and she is in the pedigree of most of your Leonbergers. So I would like to shift now to a quick overview, and you can see this in much more detail on the website, which is leogen.org, on how we are proposing to do this. So this will be a multi-generational collaboration between 3 Leonberger breeder teams located in Britain, Finland, and Switzerland. The first generation will be our carefully selected genetic donors and will result in, there will be 3 of them, 3 separate ones, and they will result in 50-50 or F1 generation crosses. Selected females will be rotated around the circle of breeder teams. The remaining puppies will be placed in carefully selected guardian homes. Guardian home puppies and selected puppies will all undergo health screening and selection so that they may be brought back into breeding if possible in the next generation. No progeny will be released from this program to outside breeders. And I want everybody to understand that there will be no LEOGEN doodles for sale anywhere. The second generation will see the 50-50 or F1 Leo mixes back-crossed with full pedigree Leonberger stud dogs. This will result in a generation that are genetically 75% Leonberger plus 25% the outside source. Progeny will again be rotated around the breeder teams, assessed, evaluated, and safeguarded in the same manner. The third generation will see them cycled around the teams and crossed amongst themselves using the 50-50 F1 males from the first cross. This will result in a generation that are still over 60% Leonberger, plus contributions from 2 outside sources. Again, selected individuals will be shared between the breeder teams, and all of them will be evaluated according to their genetic profiles as well as their phenotype. The fourth generation will see them crossed with the remaining males from the 50-50 F1 generation. And this will result in progeny that are still over 50% Leonberger, but who now each contain contributions from 3 other sources. The final step is to backcross the selected individuals from this fourth generation with full Leonberger males, which will result in a population of individuals that are nearly 80% pure Leonberger, but also contain desirable contributions from 3 outside sources. And most importantly, they will be accepted into the stud books by the registry bodies and will therefore be available to the Leonberger breeders of the future. And when they are crossed with Leonbergers, the results will be nearly 90% or over 90% pure Leonberger. But I want to stress one thing here: if they choose to use them. And here it is immediately worth stating clearly that no one will ever be expected at any time to use any progeny of the Leonberger outcross project. So in summary, at each stage, selection will be made based on confirmation to the Leonberger standard, character assessment, and on the tracking of those desirable DNA traits that we will be introducing. So will it work? We don't know. And we won't know for at least 12 to 15 years. What we do know is that the science tells us that a single outcross, then subsequent backcrossing, will be next to useless in a handful of generations, and that to make a true difference, more is needed. Hence the 3-team model. And in fact, it's worth pointing out here that a recent scheme involving cavaliers has been approved in Finland, which uses 5 outside sources. And that's about to start. So let's talk about type. Many people who oppose this proposal do so because they are quite certain that it will ruin the type of the breed forever. So it seems sensible to take a few minutes to speak about what precisely type is. The pedigree dog world is based on written standards that describe the external appearance, movement, and behavior of the ideal for that breed. After all, this is all that the judges can assess when a dog enters the ring. They are concerned with phenotype, what a dog looks like, not genotype, what genetic material goes into it. One could argue that this is precisely for this reason that many registry bodies have mechanisms already in place for allowing the inclusion of individuals of unknown origin, should they be properly assessed as being of sufficient type. For example, the UK Kennel Club requires evaluation by 3 championship status judges, and if the individual is found to sufficiently conform to the standard, then this allows entry in their registration database under the so-called asterisk system. In other words, they will have an asterisk next to their name they can be bred from, progeny will carry an asterisk, and so forth for the 3 generations, and then they will be considered pure, full members of the breed. The French Kennel Club also has its Titre Initial scheme, which similarly admits individuals of unknown pedigree into the breed lists. And there are other similar schemes. And let's face it, there have been many stories about unauthorized crosses and dubious parentage claims over the years in our breed, which is why the so-called clear by parentage status for DNA testing does not extend beyond a single generation. And has any of this ruined the phenotype of our breed? Will the introduction of dogs from outside of our carefully guarded closed stud books produce nothing but useless mongrels that will never have any qualities needed for respectable breeders to consider? Will we ever be able to regain type? This is Bindi, and she is not a Leonberger. DNA analysis unequivocally indicates that she is a German Shepherd crossed with a Saint Bernard, with a dash of Golden Retriever thrown in for good measure. I have spoken with her owner, who kindly gave me permission to share this image. She also shared her DNA analysis report with me. And she reports that Bindi is a big girl with a kind heart. I defy anyone to say that she does not, at first glance, look like a Leonberger. She has a strong rectangular head with clearly defined stop, good broad muzzle, darkly pigmented and reasonably tight lips. Her brown oval eyes are kind and expressive. Her black mask rises to just above the level of the eyes, and her well-feathered, medium-sized pendant ears, although perhaps slightly high-set, are used expressively and hang down correctly to frame her face. Her coat color and quality, her overall size and presence, calm and confident demeanor, etc., all present as a Leonberger. One could say that phenotypically, she is a Leonberger. She may not win her classic crops, but crossed back to a good quality male, her offspring might. This leads us back to the key point once again. The concept of pure-blooded pedigree dogs was invented in Europe approximately 150 years ago, during the Age of Empire, at a time when eugenics was a popular concept and when the classes within human society were strictly segregated. Although it is true that within the wider realm of livestock production, there have been breed types for many centuries, any stockman, however, will tell you that trying to sustain a population by repeatedly using only 20% or less for generation after generation will lead to disaster. Renewed strength in a breeding line comes from an infusion from the outside. If we want to see our breeds survive in a healthy form for generations to come, then we cannot continue to follow the model of dog breeding invented by those Victorian gentlemen 150 years ago. It leads to a genetic dead end eventually. We all must find a new model if we want pedigree dogs to thrive. So I want to add a few more additional points here. What LEOGEN is not about. This proposal, if it goes ahead, and remember, it's just a proposal, will change nothing about how current Leonberger breeding operates. No one has to take part. No one ever has to use the progeny of LEOGEN if they choose not to. And we are not trying to change or recreate the Leonberger. So let me use the example of another scheme to illustrate how we're different from some other approaches that are out there to preserve diversity. So some of you will have heard about the Austrian Leonberger Club, which is working with a company called Farragen, who do DLA array assessment schemes. Testing scheme. So remember, the DLA is a portion of the genetics of a dog that relates directly to its immune system. The more diversity in these immune system haplotypes, the more robust the individual's immune system is assumed to be. Farragen have identified something like 7 DLA haplotypes in Leonbergers, 7 or 9, depending on which generation of documents you read. There are estimated to be more than 200 DLA haplotypes in the wider canine population. Farragen's project is aimed at retaining as much as possible of the current diversity that we presently have in the breed, which is a very important thing to be sure. In order to do this, Farragen's project relies on as many breeders participating as possible. The more that use their test, the better the results will be and the more successful it will be. And that is a good thing. It is an excellent tool for preserving the current diversity if people use it. LEOGEN's strategy is not the same. It is the opposite. However, we believe it is complementary. We are concerned not with retaining as much as we can of what small diversity we still have. Instead, our effort will be aimed at bringing more diversity into the breed. Furthermore, LEOGEN does not rely on the participation of any outside breeders. We already have enough support. No one that does not wish to be involved needs to be involved. It will not impact any breeder in any way if they do not choose to become involved. No one, as I said, needs to use any of these dogs if they do not wish to, ever. So, as previously stated, it is not our intention to bring about change in the phenotype of the Leonberger. Quite the contrary. We are doing this because we love the breed, and it is the very points of the standard that define what a Leonberger is. Therefore, it is fully our intention as much as possible to honor the standard by selecting for type at every step and to not impose any changes on the breed. If we are successful, of course, our goal is to see LEOGEN dogs in the Ring of Honor 15 years from now based on their own merits, their phenotype. But it is also our sincere hope to see those same dogs routinely living past their 10th birthday. As illustrated by the bobtail boxer examples earlier, experience in other breeds has shown us that type can be regained, maintained, and preserved during the course of similar outcrossing projects. Many of the top Scandinavian boxer lines today, I'm told, go back to the progeny of that project. Success for our scheme, if it goes ahead, will not demand any compromise in quality or type or breeding practices from anyone else. And again, I want to emphasize, any breeder who does not want to be involved does not wish to. There are an average of 1,800 litters of Leonbergers born worldwide in any 3-year period. If we do this, we may, if we're lucky, produce 3, or perhaps, if we're very lucky, 6, if we're wildly successful during the same time frame. 6 litters against 1,800 is not going to wreck the breed. Current breeding will carry on just as it is now, and any future progeny that we produce will succeed or not based solely on their own individual merits. However, if things continue the way that they are at present, in years to come, when the breeders of tomorrow are desperately seeking alternatives to the genetically limited populations that they will be faced with if things don't change radically now, we want those alternatives to be there, but it will be their choice. So, is this some sort of a secret project? It has been suggested that this is a secret of some sort. No, it's not. Unlike the Bernese Mountain Dog Vitality Project, it is not our intention to conduct this work behind closed doors. This is why we started with the national kennel clubs, the registry bodies themselves, and why we sought the direct support of 3 prominent national Leonberger clubs, why we published our website over 8 months ago now, with full disclosure of who is involved and why we are making presentations such as this one now. We know that a project such as this can divide people's opinions deeply, but we sincerely hope that enough breed enthusiasts can now see that we cannot simply carry on with the course that we are presently traveling. As I said, as breeders, we must get our heads out of the Victorian age, when breeding the best of the best and throwing out the rest was a recipe for success. It's not anymore. We're at 50 generations of Leonbergers, and this method no longer ensures anything other than a continued genetic impoverishment of our breed. Pedigree dog breeding, in this matter, is not cyclical. Instead, it is linear. In other words, we can't keep breeding the same way and keep depleting the genetic diversity and then somehow assume that we are going to hold our place as a breed. We're not. Instead, if things do not change, there will be a continued march towards the cliff of genetic calamity. The science clearly shows us this. The kennel clubs now recognize this. The breeders of previous generations saw this, but is now up to the breeders today, as custodians of this lovely breed, to recognize this and to take the steps necessary to change things. And finally, we are sometimes asked, what's this going to cost and who's going to pay for it? And yes, this is going to be a long project, 12, if it happens, 12 to 15 years, we predict. I, for one, may not see the end of it. We know that we cannot rely on the participating breeder teams to fund this, and we will not be producing puppies for profit to cover the costs either. Therefore, fundraising will be necessary, and indeed it will be vital to the success of the project, and it is therefore part of our ongoing planning to put a fundraising strategy into place once we know we are going to proceed. Academic grants will also be an avenue worth exploring because of the long-term implications of such a project as a study. And so that's pretty much brings me to the end of what I was going to say. And I'd like to hear anybody's questions if you've got them. Thank you.